So yeah, my name's Chris. I'm a biostatistician in the Department of Biostatistics at Vanderbilt Medical Center. A biostatistician is just someone who uses statistical methods to derive inference from biological or biomedical data. So that's kind of the angle that my talk will take today, but I hope that it has a little bit more general appeal than that. We've already heard about fraud a couple of times this morning, and I'm going to at least motivate my talk with a little bit more fraud. In 2006, in Nature Medicine, one of the more prominent biomedical journals out there, this paper came out by Anil Pati et al. From Duke University, and it claimed to be able to use what are called genomic signatures, which are essentially hundreds of genes acting together to guide the use of chemotherapeutics. So this was a lung cancer trial, and they purported that people had these unique signatures in their DNA that allowed them to predict how well chemotherapy drugs would work or would not work on individual patients. This is sort of what you may have heard of as personalized medicine. And so other scientists were very excited about this and tried to reproduce it. In particular, some folks at MD Anderson in Houston tried to do this and failed. And so they started looking a little bit closer at it and started finding that the story was unraveling bit by bit. And the problem with all of this was not so much that, well, it turned out to be an enormous fraud. So initially it seemed to be a lot of carelessness. There were a lot of off-by-one errors. There were references to columns in a data frame that were, off by one. And there were transposition of labels. So individuals that were labeled as sensitive were those that were resistant. Very careless, seemed like clerical errors. And as they dug deeper, it was proven to be a lot of it was proved to be completely made up. And the problem though is that it took hundreds, actually more than 1,000 hours between these two gentlemen here, Keith Baggerly and Kevin Coombs, to discover this. Otherwise, it may have been published and received the attention that it didn't deserve, and folks would have tried to build on that, essentially leading science down the garden path. So what I'm going to talk about a little bit about today is reproducible research and how some simple tools, particularly one in Python called iPython, can help us make our research more reproducible. And essentially what reproducible research is being able to reproduce the conclusions from an experiment based on the measurements from that experiment. So not every experiment is exactly reproducible, right? If you go and reproduce an experiment in another lab in another place, things will be slightly different. But given a particular data set, you should be able to retrace your steps from the conclusions of your analyses back to that data set. And this is actually pretty hard. Even using your own work, I've often had to go back to work that I've done 2 years prior and try to reproduce it or recreate it in some form in order to extend it and build upon it. And really the most basic form of reproducibility is simply a description of your data, the methods that you used to analyze it, and hopefully the code that you used to produce those results. And when you read a journal article in a scientific journal, hopefully they've at least provided that description. But these days, scientific data are becoming very large, very complicated, and the models that we use to analyze those data are becoming even more complicated and larger. And so simple descriptions simply aren't adequate for this reproducibility. And as a result, most of what you read in journals and hear about on the news is not reproducible. It would be very difficult for you to go back, even as a scientist, let alone a private citizen, to go back and essentially forensically look back and see if the conclusions derived from a particular data set are valid or not. So you may be surprised to find out that reproducible research simply isn't part of the current culture of doing science, and certainly not in computational statistics. And one of the, I think, one of the more innocent reasons, and this isn't just people being nefarious, these are folks that are doing genuine science and generally being very diligent about their research. But there are a lot of impediments in the scientific computing workflow that keep us from being reproducible. For example, you may start out doing some exploratory work in a shell, perhaps, in a Python shell or an R shell. If you're a statistician, you may be an R user. And that may lead to some collaborative development with other researchers that may or may not be statisticians, certainly probably aren't programmers. And then if that's successful, you move on to production, publication in a journal, and then communication like I'm doing now, presenting perhaps at a meeting. And at every interval between each of those steps, there's kind of a break in the chain. In some cases, you start from scratch again. Particularly, going from production, you may forget about the work that you did back in the exploratory stage where you may have discovered some things. And a lot of the standard tools impose some of these barriers. But very recently, among the Python community, there's been a development called iPython. And iPython began very modestly as simply an enhanced shell to Python that provided a lot of programming niceties, better exception handling and display, better debugging tools, some nice shortcuts, colorization, things like that. And it's become a very much more robust productive development environment for Python programmers. And in particular, of note that I'm going to focus on today is what they call the Python notebook. And it's simply this HTML notebook that I'm using right now that allows us to integrate not only code, so each of these cells is just Python code that we can run. Oops. There we go. Simply plotted a line from a function and augments it with HTML and Markdown annotation. So we can add some headings in here. There's just plain Markdown. And what this allows us to do is, as we're working on code, to annotate it, to describe it, and as a step towards making our work more reproducible. The iPython notebook simply consists of a couple things. One, this whole notebook is just a JSON file. So you can store it on GitHub and distribute it very easily and display it very easily. And then a web-based user interface that you're seeing now for doing the authoring and for running the notebook documents. And all you do is you start it up in a console. So rather than, usually when you're using iPython, you just say iPython and it pops the interpreter up. iPython notebook starts up an iPython engine and an iPython controller that allows one or more clients to connect to the engine and start running Python jobs on it. And this notebook is great because it provides everything that you expect from a browser interface. So for example, you can embed an entire web page in a little frame here. So we're importing this HTML module from the iPython display, and I've just loaded up the page for this meeting. We can embed some video, so we just take the YouTube identifier and pop that in. It runs inside of the browser like so. Remote code can be loaded interactively. So for those of you who use Python and use matplotlib a fair bit, which is the plotting interface, one of the main scientific plotting interfaces, you can just go, if you're like me, you hardly ever code up a plot from scratch. You go to the matplotlib gallery and find something that looks kind of like what you want to do, like so. And what we can do is just grab the link to the source code here and go back to the console, and we'll load it in. And this will pull the code in, and this is completely editable. I can go in and annotate this and change it as I need to. Here I'm going to run it and generate the code. Okay, so it makes it really easy to integrate code from other places. Obviously, in 10 to 15 minutes, I can't give you the full iPython tour. There are actually very good videos of Fernando and Min Reagan Kelly and Brian Granger, who are kind of the brain trust behind iPython, giving various comprehensive hour and a half long talks that I encourage you to explore. But I want to hit on some of the major things that make this really useful as a scientific tool. MathJax support is integrated. For those of you not aware of MathJax, it's a little JavaScript library for rendering LaTeX equations. So as scientists, and particularly statisticians, we've got to describe our work using equations. And this is just a simple LaTeX equation here. And you enclose it in a couple of dollar signs as part of a markdown cell. And it renders quite nicely the equation. It supports a lot of really good scientific third-party packages. One of the nice ones being SymPy, which is a library for doing symbolic math. So that what we can do here is we load this SymPy printing library here. So %loadExt is the way that we load an extension into iPython. And so we're just going to create a couple of variables, x and y. And then this just defines a polynomial equation. So we've got x plus y squared, and we're multiplying it by x plus 1. And what IPython does is it gives us this pretty printed equation the way we like it, but it's not just a pretty face. The EQ is actually that relationship, and we can do things like expand that. If I want to take the first derivative, I can go in and do that. So I'll take EQ with respect to x, and that's the first derivative of EQ. We can generate Taylor series just like that. Okay, really, really nice. Magic functions is probably the killer feature of IPython, which I already used once here by loading this extension. There's a whole bunch of predefined functions that come with IPython for doing things like debugging, loading extensions, and so on. You can get a whole list of what they are here, including being able to integrate other languages into the IPython notebook. Just as an example, we can time the execution of our code here. So I've got some eigenvalue calculations done 2 different ways, and it'll go in and run it 100 times or so and calculate how fast it's taken in each case. Embedding interpreters can be done here, and these ones are popping up all the time, so we can insert some Ruby code or a Bash script. For those of us that are statisticians, at Vanderbilt Biostatistics, it's essentially an R shop. I'm kind of the Python black sheep of the family. And so I have to use a lot of other people's code quite a bit, and there's a lot of really good code written in R. So what I can do here is load what's called the R magic. And so in this line, I'm generating some fake data in Python. So these are going to be NumPy. These are going to be NumPy arrays. So if we look at x and y, that's what they are. And then here's some R code for doing just a simple linear regression. So it's going to take my x and y Python input, do some stuff in R, particularly a linear regression and a plot and output of the coefficients of that model, and then send it back to me and send those coefficients back to me for later use in Python. So if I run this, this is just R output, for any of you familiar with R. And most importantly, those coefficients are now in the form of a Python list. So it does all of the translation from an R data structure into a Python data structure. So it's really, really nice. As a sort of more detailed example of that, this is some of the work that I've done in my previous life as a wildlife biologist studying manatees. So manatees are those Sirenian marine mammals that paddle around off the shores of Florida. We want to be able to estimate how big the population is because it's an endangered species and so forth. And of course, a lot of the folks that do wildlife biology also use R. So I've got a whole bunch of simulated data that a colleague did for me here in R, quite an extensive simulation. And so I'm taking their simulated data, fitting it in my Python model, and seeing how well my model does. And so this simulated data returns on our data frame, and we rather seamlessly here go take the output from that survey data and turn it into a pandas data frame, which we can slice and dice in Python really easily. One of the other nice things about iPython is how easy it is to turn things parallel. And you may or may not be aware that doing parallelization in Python isn't always easy, not right out of the box anyway. And so one of the nice things that iPython does is it makes parallelization pretty straightforward. This is sort of the iPython notebook console. Here's a list of a bunch of notebooks that I've worked on. And you'll see one of the tabs here are clusters. And so my sad little air here only has two processors on it. I think it's got 4 virtual ones. But I can easily just spin up a couple of engines here, like so, and go back to my notebook and import from iPython Parallel a client that will do all of our parallelization for us. Oops. Sometimes it takes a second to, there it is. Okay, so now I've got 2 direct views to each process, and there's just obviously a silly little function that's going to tell us which process it is and what host that it's on. And so we've just done just like that. For me, for someone who isn't really good at parallelizing things in Python, this is a boon. Okay, I'm a scientist first, I'm a computer scientist second, if at all. So being able to do this is really, really nice. So where does this all fit in with reproducibility? Why is it such a good tool? Well, this is some work that we're currently doing. This example is a model of a very large database of children with hearing impairment. And it's essentially how they progress on tests of articulation, receptive vocabulary, and so forth, as a function of a whole bunch of covariates. What their mother's education level is, their socioeconomic status, how old they were when they found out they had hearing loss, what sort of technology are in their ear, and so forth. And so I'm working with not only non-technical scientists at Vanderbilt, but a lot of the people that will be reading this stuff are going to be school administrators who certainly aren't going to know what I'm doing with my code. And so using the notebook, I'm able to pull in data that they've given me in a spreadsheet and do quite an extensive cleanup of the data set. In fact, here, this is just from an Excel file parsing 4 different sheets within that, spreadsheets within that file, and then merging them all together again. So I'm going through and I'm cleaning things up, I'm recoding variables, and at each step, I'm describing what I'm doing, okay? So that even though the school administrator doesn't know what all this is doing, I've told them what they're doing here, and then I'm displaying something. I'm displaying the head, the first 5 rows of the cleaned up data set, and they may say, oh, well, this isn't right. I know that individual, they're not supposed to have premature equals 2, and so on. They're able to drill down a little bit and look at it and validate it somewhat. So I can do an extensive cleanup here. I can display some summary graphics. This is a histogram of the age distribution in months of individuals in the database. It's like 4,000 kids. Demographics, what language they speak, the age at which they had amplification, and so on, all the way down to me building a statistical model and merge the data sets. And I build a statistical model here in Python. that's able to estimate the relationships between those data, or between those variables, and see what effect those variables have on how well these children do on their tests. And so what the results that the, and here's a full description of the model, all in the same document. And then this is what the administrators want to see. So, you know, being premature is negatively associated with your test score. Being male slightly improves what you do on the test score. Having a non-English language as your primary language has a very negative effect on how you do in the test score. And they're able to leverage this database, all this data they've been collecting, and not knowing what to make of it. A really nice thing is that we're able to distribute this in a couple of different ways. I can, because this is just a JSON, I can post this, you know, on GitHub, which obviously you can't view it here, but it's really easy to render this in HTML. So this is for a different analysis, but there's a notebook viewer that turns that into a static HTML file. This is just HTML. So I can pass this link on to a collaborator. They can look it over. This is for a measles outbreak analysis in Brazil. And you've got a report in HTML like so. There's also a converter, NB convert, that takes a iPython notebook file and converts it to one of several formats: LaTeX, Markdown, HTML, restructured text, what have you. And so what that allows me to do is to turn that HTML or that notebook file into a PDF, for example. Taking it to its extreme, one colleague is using the iPython notebook to write a textbook. Okay, so this is a textbook on doing Bayesian analysis in Python. This is just an HTML. This is just an iPython notebook, and it of course contains equations, code, and so on. And using that NB converter, we turn it into a product like that. So you go from hacking, you know, opening a new blank notebook, like so, start hacking, something works, start annotating it. And so you can go all the way from there to a LaTeX file that you could submit to a journal for publication rather seamlessly. So it takes all of those pieces of friction out and gives us fewer excuses not to be reproducible. As a sort of a gold standard to reproducibility, this really nice paper by Titus Brown, which some of you may know from Twitter, on shotgun sequencing. It's a way of doing DNA cloning and sequencing. Not only has he got his archive preprint of his paper, but down here he's got his Git repo of the analysis, the repo for the paper itself, instructions on running the paper. Here's the HTML view of his iPython notebook. Let's have a peek at that. Okay, how he created figure 1, how he created figures 2 and 3. Okay, you don't get that most of the time. 8 gigabytes of data required to run the analysis. Okay, so if anyone thinks that Titus was up to no good, they can show it, right? And as a researcher, I'd want people to go in and poke holes in my research before I published it, right, and embarrass myself. For those of you who are interested in more curious, there's nbviewer.ipython.org serves as a repository for folks' notebooks, ones that they're able to share, but also a viewer. So you can enter a gist number here, or a URL that contains an iPython notebook, and it'll render it as HTML, as I showed you before. But down here is kind of a little gallery of folks' iPython notebooks, like how to make your matplotlib plots look like XKCD plots. Okay. So, exactly. A couple of these are mine. How to use pandas. It's a good place to put tutorials. How to use pandas. Everybody wants to know how to use pandas. Some GIS stuff, Google Earth. You can embed, again, almost anything in this. So, so that's really all I've got. You know, hopefully, I'm very grateful for the Python community and folks like Brian and Fernando and Min that are able to provide this terrific tool that I think is much bigger than they think it is, and it's already serving to pull some new folks into doing statistical analysis in Python that weren't there previously. So, thank you.